Hello, welcome to Being Patient. We're with Dr. Christopher Duma. He's a board certified in neurosurgery and serves as medical director of Hoag Memorial Presbyterian Hospital's brain tumor program in Newport Beach, California. He's also president and founder of clinical stage company Regeneration Biomedical. He has practiced neurosurgery since 1987. His expertise in the surgical management of brain tumors, Parkinson's disease, and hydrocephalus, and is skilled in minimally invasive neurosurgery. And he also performs brain mapping and employs his patented technology designed to halt the spread of malignant gliomas. His gamma knife leading edge technique holds a U.S. patent and allows him to halt the migration of primary brain tumor cells through the brain in a very novel way. Additionally, Dr. Duma has been performing deep brain stimulation since its FDA approval in 1998 to control movement disorders such as Parkinson's disease. And we haven't even got to the main thing we will discuss today. Dr. Duma has the world's first experience of injecting autologous, which is coming from oneself, mesenchymal fat-derived stem cells directly into the ventricles of the brain for treatment of neurodegenerative disorders like Alzheimer's disease, ALS, and chronic traumatic encephalopathy. Now, Dr. Duma, first like to ask you, you pioneered injecting this technology, fat-derived cells into the brain. Is it essentially taking your own stem cells and injecting it back into the brain?
Thanks for that introduction, Mark. That was very kind of you. Yes, this is the first of its kind in the world where we're actually taking someone's own stem cells from their fat in their abdomen, abdominal fat, supercharging it, obviously growing those cells, expanding them, supercharging them with a special supercharging agent called Wnt, which we can discuss later. It's very scientific. But what it does is it turns stem cells on and makes them very active. And that is what's injected directly into the patient's brain, the patient that has Alzheimer's disease. And you're right, we're hoping to use this for other diseases like ALS, Parkinson's, MS, chronic traumatic encephalopathy, what the NFL football players get. So we're starting with Alzheimer's disease. And the real issue is, how do you deliver a drug into the brain that makes it there and stays there and works and operates? And other companies have tried intravenous forms of drugs, oral forms of drugs, and they just don't make it into the brain because of that famous blood-brain barrier. We're trying to, thwart that little barrier and break through it by directly injecting the cells into the cavities of the brain.
And how does that even look? We're just envisioning how do you inject it in, and what sort of devices? What's the setup look like?
So, Mark, I have a picture of it. Would it be okay to show that?
Yes, sure.
Okay, so let me share my screen then here and get you a real idea of what we're talking about. So I hope you all can see that. Our brains are hollow. A lot of people in your audience have no idea that our brains are hollow. It's like a cantaloupe. If you cut the cantaloupe in half, it's hollow in the middle. Our brains are the same way. And these areas here are called ventricles, and they are full of fluid. Imagine putting a little reservoir or a port that actually communicates with that. So then in the office or at a clinical setting, you could put a needle into that little reservoir and inject your own stem cells. They go down the tube into your ventricles, and then they percolate into your brain. And it turns out that our brains are very permeable as we get older in this fashion. So we can deliver stem cells that get into the brain. We've done work with rats, with pigs, and we've already proved this type of thing. But right now, we're just finishing the phase 1 trial in humans, and the results have been very exciting.
Yeah, I think you've given us a cliffhanger there. What do we know so far in the results?
So a phase 1 trial, which is an FDA cleared trial, is designed for safety. You have to determined that since this is the first in the world or first people to ever get this type of treatment, we want to make sure that it's safe. And these patients are very brave, but they've all exhausted any other therapies. They have no other options, and they're clinically deteriorating. So they have no other options. We enroll them in the study, the trial, and we put the reservoirs in. We take their cells, we expand them in our laboratory, and then we inject them into the brains. And for the phase one, it's just a single injection. And so just to see if the patients do well. And Mark, I'll be honest with you, not one of our patients even got a headache from the injection.
Wow.
So they all did beautifully in terms of safety, right? And then any researcher, any research team that's doing this type of work, we're all interested in what's called secondary effects, secondary endpoints. Even though this is just a safety trial, did we see any patients get better? Did we see any improvements in the amyloid beta levels and the other levels of what we call phosphorylated tau? Tau protein is bad also. And for for the vast majority of the patients that we treated, their tau levels went into the normal range, their beta amyloid levels decreased, and for the majority of patients, their cognition actually improved, which is not the case with the other drugs that are out there now. The Biogen and the Lilly drugs, they do well to get rid of beta amyloid, but they don't really have a great effect on cognition.
Now you say cognition improve by what measurement and how much?
So, yeah, we can't just rely on family members to say, hey, they're doing a lot better. So we do have cognitive testing. We use various uh, tests for cognition. One is called the Mini Mental Status Exam, MMSE. Another is called the ADAS-Cog, the Alzheimer's Disease Assessment Scale of Cognitive Scoring. And so that is all, been over the years, those are the gold standard for cognitive testing. And so you can see what their baseline is and whether they're getting worse or better.
Mhm. Is there a measurement by how much though? I mean, does it vary? Did it vary quite a bit or?
Sure, sure. Good question. To be quite honest with you, if they improve by even the smallest amount, that's a first in the field of medicine. So we were very excited that the majority of our patients improved. You're talking about an improvement from, scores that might be in the 70s, and 70 higher scores are worse, and you get them down into the 30s. That's a significant difference. So we were seeing that. But remember, my disclaimer is this is not a phase two trial. This is like secondary endpoints in our phase one trial that give us hope to move to a phase two.
Okay. And before we talk about phase two and what lies ahead, what type of patients did you test this on? Is there a certain
That's right.
age group
That's
or
right.
stage that you went with here?
Absolutely. So there are seven stages of Alzheimer's disease. So one through seven. Stage three is called mild cognitive impairment, MCI. Uh, Donanemab and lecanemab, the Lilly drugs and the Biogen drugs, they use those patients in their series. We did not. We only used stages four and five. So mild to moderate Alzheimer's disease were included in our trial, and those would be the same level of severity used in our phase two trial as well.
Was there a certain age group that you target, or was it a variety of ages?
Right. In the phase one, we used 45 years old to 80. And based on that, in the phase two, we're going up to 85.
Okay. So you're talking now phase two. Will that just expand to a wider pool, or tell us about phase two?
Sure. Sure. So it's an exciting week, actually, Mark. On last Friday, we were actually cleared by the FDA to go to the phase two. So we presented our data to them about a month and a half ago. and from the phase one, and they reviewed it and they allowed us to move to the phase two at this point. So phase two enrollment is beginning very shortly, probably within the next couple of weeks. So that's extremely exciting news for us. The phase two is an efficacy trial. So we're seeing, does this really work? Okay, we had a clue in phase one that it was working, but is phase two that will actually tell us whether it's working statistically. So for that reason, I'll give you an example. In phase one, we have only 9 patients. In phase two, we're going to have 115 patients and 115 patients spread around the United States. So we're going to have 5 sites as far as Florida, Northern California, Southern California, and that will be a multi-institution trial. And so that gives us a much broader idea as to whether this really works or not.
Are you targeting a specific type of Alzheimer's? I mean, what particular are there best candidates for this particular testing?
Yeah, we are. We're targeting all forms of Alzheimer's disease. We are excluding Pick's disease, disease related to Parkinson's disease, like Lewy body dementia. And so there is an inclusion and exclusion criteria for our trial. And the neurologists who refer and our principal investigators make those decisions whether they're appropriate for the trial.
And how does it work for the uh, uh, test? Whoever's part of the trial, do they have to come back or regular sort of treatments for it to tell us about how that works with the particular?
Yes, as you might imagine, we do follow-up exams, we do follow-up MRIs, we do follow-up PET scans. And this is all paid for by the company, Regeneration Biomedical, that's sponsoring the trial. There is zero out-of-pocket expense for the patients, which is wonderful, because it is an FDA trial. And so that, yeah, they have to be actively followed up with various scans and even clinical exams. So every 3 months, for example, they have to come back for an examination. Every month and a half, they might get some more blood tests done, spinal fluid drawn, that type of thing. So we have a full regimen as to exactly when they would be coming back. But the phase two is only injection for a year. And remember, I told you phase one was a single injection. Phase two is an injection every 2 months. So they would get the cells injected into the brain every 2 months in a clinical setting.
Okay, we've actually got a question from one of our viewers, and they're asking, are ApoE4 homozygotes included or excluded?
Right. So the reason that that patient or that viewer actually asked that is because for the two new drugs, no one likes to do them in the APOE4 population because they have a higher risk of brain hemorrhage and brain edema. We do not exclude them at all. In fact, we embrace the APOE4s. So reach out to us to get into the phase two.
Okay, we've
Now,
got
for the rest of your audience, an ApoE4 gene allele, everybody gets one gene from their mom and one gene from their dad, right? So you could be, the gene is APOE, and one of them is called a 3 and one of them is called a 4. And if you have a double dose of 4, you have about a 60 to 70% chance of getting Alzheimer's disease as you get older. It's enormous. If you have a 3-4, one 3 from your parent, another 4, you have probably a 20 to 30% chance of getting Alzheimer's disease. So that APOE4 allele is very important in making the diagnosis, telling people what their chances are in the future of getting this disease, but also whether you're going to get the treatment from the drugs that have just been FDA approved or not. But we're open to all. We'll take a double 4, no problem.
Okay, we've got some curious watchers out there. Here's another question. How does plasmapheresis treatment currently being used for Alzheimer's relate to the work you are doing, or does it?
It doesn't relate at all. Plasmapheresis has theoretical, benefits where you're getting rid of the toxins and the bad things in your body that are building up. You definitely will be getting rid of some beta amyloid that way. You'll definitely be getting rid of some phosphorylated tau that way. But as I said, is the stuff floating in your blood the bad stuff, or is that just the debris from, you know, from the cells not doing their thing properly? I have this great slide that I show people, and I think it would be great to show it right here because it really burns this into your brain as to what we're talking about. So this is my slide. If you have a refrigerator and it's producing spoiled food, why would you keep removing the spoiled food instead of fixing the refrigerator? So our theory, our hypothesis is that you have to fix what's going wrong in the brain, what's going wrong, instead of just remove the amyloid plaque or amyloid beta with a monoclonal antibody. So you got to get to the source of the problem instead of just keep removing the end products of cell death. So that's the philosophy of regeneration biomedical. And that's where my whole thinking was coming from.
Got another question from Barbara. There may be some folks in the ApoE forum who are interested in more info. Where can they go to find more info on phase two?
I don't know whether you're going to show my website address, but regenerationbiomedical.com, all one word, regenerationbiomedical.com. And the link for that is, they're all in there. You go to the clinical trials section and you'll see the ongoing trials.
Okay, another question from a viewer. Is there any research about use of stem cells for non-diabetic peripheral neuropathy?
It's a good one. Boy, everybody would love the treatment of non-diabetic neuropathy, peripheral neuropathy. It remains to be seen, to be quite honest with you. What's going on with neuropathy is those sensory cells in your feet and your hands, they're dying off. Perhaps some stem cell work would help for that. Right now, we're not focusing on that. The next diseases we're going to move into are chronic traumatic encephalopathy with the NFL players and probably Parkinson's disease or ALS. But we need funding for that. We need large numbers of money for that. So we will move forward depending on our resources. But right now we're funded for Alzheimer's disease. We're moving in that direction. Interestingly, I'm speaking to the NFL people today after this talk. So maybe they'll be all interested in putting their money together and helping their athletes.
Okay. Well, what milestones would convince you that cell therapy is sort of ready for wider clinical use?
Yeah, I think the phase 2 trial will tell it all, Mark. We have some very, very promising clues from the phase 1. So much so that I wouldn't go to phase 2 if we didn't have these clues, right? I mean, if we had bad side effects or people were getting sick from the injections or it didn't do anything at all to any of the biomarkers. I mean, we look at spinal fluid biomarkers, spinal fluid CSF levels of beta amyloid and phosphorylated tau, and I told you that the majority of patients, they all responded in the positive direction. So those are the suspicions. Now with a phase 2, we will know whether this works or not, and then it will move on from there. I would hope that I've opened the door for more research of injecting things directly into the brain. A lot of people with cancer, they get a little port. It's called a portacath, and it's put in the vein. It's put in a subclavian vein under your collarbone. And that's so you don't have to keep getting IV in your arm. You can poke a needle into that and get your IV chemotherapy much more easily and pain-free. Well, we have the exact same thing for the brain. People don't know it. I did not invent this. It's been around for 50 years. It's called an Ommaya reservoir, and we put it in, and the use of it was for chemotherapy. But I decided to use it for stem cells.
And this work stems from you, treating Alzheimer's patients traditionally, and sort of seeing how a lot of things just really were not that effective.
Yeah, 100%. That's my mentality. I'm a neurosurgeon. I see patients with dementia. I see patients with Parkinson's disease walking into my office, and I've been doing it for 30 years, Mark, and there really haven't been any major strides in any of these diseases. So I sit back and I go, what shall we try? What should we do next? And that's what drove me to do the rat work and the pig work and then go through the FDA. By the way, the stem cell industry has a very bad reputation. I mean, there's a lot of snake oil sales that are going on out there for with stem cells. There are rogue clinics that are out there in Mexico and other places that tout the safety and the efficacy of their stem cells, but there really has been no FDA trial for things injected into the brain. And in order to legitimize this, I decided let's go through the FDA.
Okay. I think you've answered all my questions. I just wanted to leave you with any final words about the future and of treatment and whether you believe or are confident that stem cells will play a greater role.
Yeah, I'm not going to say 100% confident. There's no 100% in my field, but I am 95% confident that we'll be using stem cells specifically for these neurodegenerative disease, which we've had nothing for ever. So it is a new prospect. I can't wait to see the results of the phase 2. And as I said, we just need funding for that to move forward. So the sky's the limit, to be honest with you, when it comes to stem cells.
Okay. Chris, thank you so much for joining us and sharing your insight and some fascinating work. And thanks to our audience for tuning in. Don't forget
Good
to sign up
questions,
for our
yes.
newsletter on beingpatient.com, where we'll keep you up to date on all the upcoming talks. Thanks again, Chris. See you next
Thank you
time.
very much, Mark.